Today we're talking about how different types of lymphocytes are going to behave in the immune system. We're going to spend most of our time talking about specific immunity or adaptive immunity and what two classes of lymphocytes are going to do when encountering a previously encountered antigen. And we'll also discuss the natural killer cells and how they are interacting with the rest of the system. So of course we do have three general types of lymphocytes. We have our T cells, our B cells, and our natural killer cells that kind of go over here in the corner. Now, they're all lymphocytes. They all basically look like a lymphocyte. They've got a large nucleus. They're agranulocytes, scant cytoplasm. There's going to be very few changes to this overall pattern, so the illustration is maybe not the most useful part until we get to a couple of details in here in the B cells. So the T cells can differentiate into two different types of T cell. We do have helper T cells or helper T lymphocytes, and then we also have cytotoxic T lymphocytes or T cells. So helper T and cytotoxic T. Now, the way these two behave, they do interact with each other. They are both necessary for immunocompetence. This is going to be our cell-mediated specific immunity. We're also going to have two different kinds of B cells. We're going to have memory B lymphocytes. And they're actually going to undergo mitosis into more memory B lymphocytes, as well as a special cell called a plasma cell. Now, we can illustrate a memory B lymphocyte a little bit differently if we would like to. That memory B lymphocyte tends to have some specialized antibodies that are integrated onto the surface of this cell. So we'll get into the actual mechanisms and what's happening here in just a moment. Let's start back over here with our helper T lymphocyte. So our helper T lymphocyte is going to be able to recognize and bind with major histocompatibility complexes. Specifically, they're going to interact with class 2 MHCs. This is, by the way, the cell that has the CD4 receptor. So we got a CD4 receptor capable of recognizing class 2 major histocompatibility complexes. And what's going to happen is that this helper T lymphocyte, if it encounters an antigen on an infected cell or a pathogen itself, it is going to do a couple of mechanisms. Mainly, it's going to release cytokines. So what is a cytokine? A cytokine is a chemical signaling molecule. It's going to increase inflammation. It's going to do a few things. It's going to be required. Cytokines. Cytokines are going to be needed for co-stimulation of cytotoxic T lymphocytes as well as B cells. It's also going to activate some phagocytosis through various macrophages, and it's also going to help create some memory B cells, memory T cells. Not previously mentioned in this cascade. So you need cytokines in order to essentially start the rest of these cells going. I always call cytokines like a call for help. It's like saying, hey, you guys, get over here. It's calling the cytotoxic T cells, the macrophages. It's calling the B cells over to the place where some infectious agent is located. It's the hey, you guys moment, right? So, hey, you guys. I am kind of channeling the movie Goonies just a tiny bit. Okay, so cytokines is co-stimulation for a variety of other things. Cytotoxic T cells are one of the things that will be co-stimulated by cytokines. You know what, let's change our arrow color so that we can have something associated with cytokines. I'm going to use this cool pink right here. Cytokines are going to be necessary for activating cytotoxic T lymphocytes. I'll give myself a little space here and show you that it's also going to be necessary for co-stimulating B cells. So I'm going to head back on over to our cytotoxic T lymphocyte. Our cytotoxic T lymphocyte is, again, cell-mediated immunity. This is a cell that is directly capable of causing cell lysis. It's going to do this with a few different tools. You may recall from talking about natural killer cells over in innate immunity that they can utilize exocytosis of perforin molecules. And what that means is we can essentially make a hole in a plasma membrane or in part of a pathogenic, either a pathogen or an infected cell. We can essentially make holes in it. So the cytotoxic T lymphocyte is also going to utilize perforin molecules. So we can have perforin. We can essentially stab a hole into a foreign cell or an infected cell or a tumor cell. We can release some other specific cytokines, and that's going to actually initiate apoptosis. Now, you should know what apoptosis is. Apoptosis is programmed cell death, so this is a non-inflammatory cell death. This is a self-destruction of a cell that is otherwise harmful to the body. So apoptosis is generally considered a desirable outcome for an infected, damaged, cancerous cell, for example. There's also something called lymphotoxin. So we can disrupt some metabolic pathways in some of our target cells here. So this, again, cell-mediated immunity means that this cell can directly attack pathogens. You know what, let's draw a little monocyte over here. This is not part of specific immunity. This is part of innate immunity, but it's also triggered by cytokines, so macrophages are also activated by cytokines, and I want to make sure that you're including that concept. Okay, so cytokines also worked on our B cells. Our B cells undergo mitosis and create memory B lymphocytes that retain antibodies for a specific thing. So what we're saying is we've already encountered this pathogen before. We already have this pathway. Upon encountering it again, we'll have mitosis of more memory B lymphocytes that is going to increase our number of sort of standard having this around memory cells, so that the more times you're exposed to a specific antigen, the stronger your immune response to that antigen can be. While it's undergoing mitosis into memory B lymphocytes, it's also undergoing mitosis into very, very many, larger number, of plasma cells. And plasma cells have a specific job of secreting antibodies, immunoglobulins. So the ones that we've been talking about kind of in the background this whole time we have antibodies for specific antigens. Now, these antibodies have a lot of different functions, and those different functions are kind of a topic for a different video, but they can directly or indirectly attack or signal that something needs to be destroyed, right? We can attack them directly with our antibodies, or we can signal other things to destroy utilizing antibodies. So this is going to be your antibody mediated immunity. So if T cells are responsible for cell-mediated immunity, B cells are going to be responsible for your antibody-mediated immunity. And of course, it's the same cytokines that are triggering all of these different pathways for destruction of a pathogen. They all need to work together. So I've kind of tended to teach this as a step-by-step cascade, where we have helper T lymphocytes first, followed by cytotoxic T lymphocytes, followed by mitosis of B cells into plasma cells, followed by antibody-mediated immunity. Now, those antibodies actually kind of come back to the start, because one of the things that those antibodies do is they signal for even more helper T lymphocytes, cytotoxic T lymphocytes, and mitosis of more B lymphocytes as well. So it's kind of an ongoing cycle, and all of these results of cytokine activation are occurring simultaneously as well. But if it helps to tell yourself a story, then go ahead and tell yourself a story. Let me see. I took a couple of notes to make sure that I don't miss anything. Trying to see if there's anything I forgot about B lymphocytes. In case I didn't mention it, they do respond to class 2 major histocompatibility complexes, but I think that's about it. Now remember that natural killer cells are off over here in innate immunity. So they are, another thing I've missed, CD4 for helper Ts, CD8 for cytotoxic T lymphocytes, and then this is all specific immunity over here. This is innate immunity over with natural killer cells. So again, they are going to attack a wide variety of infected or possibly cancerous cells, anything with an abnormal MHC, which again, that MHC is based on the genetic code. So if your genetic code changes, your MHC can change as well. Ways that your genetic code can change are through things like infection or mutations that the same mutations that lead to cancer can mutate the MHC, which is how the natural killers can recognize mutated cells at all, including cancer. Pretty cool mechanisms. I hope this helped. Let me know if you have any questions.